Stomata, singular stoma, are tiny pores predominantly found on the epidermis of plant leaves, though they can also appear on stems and other green parts. They are crucial for a plant's survival, primarily regulating gas exchange and water loss. Stomata cell structure. A stomatol complex is typically composed of three main parts: stomatol pore, aperture. This is the actual opening through which gases, carbon dioxide, oxygen, and water vapor move in and out of the plant. Guard cells. These are specialized epidermal cells that surround the stomatal pore. Their shape and turgidity, internal water pressure, regulate the opening and closing of the pore. In dicot plants, guard cells are typically kidney-shaped or bean-shaped. The inner cell wall, facing the pore, is thicker and less elastic than the outer cell wall. In monocot plants, especially grasses, guard cells are often dumbbell-shaped with narrow middles and wider bulbous ends. Guard cells contain chloroplasts, allowing them to perform photosynthesis and produce sugars, which influences their turgor. Subsidiary cells, accessory cells. These are specialized epidermal cells that surround the guard cells. They differ in shape and size from the other epidermal cells and support the guard cells in their function by facilitating water and ion movement. Not all stomata have distinct subsidiary cells.
Composition of stomata. The cells making up the stomata, guard cells, and subsidiary cells are fundamentally plant cells composed of cell wall, a rigid outer layer primarily made of cellulose. The arrangement of cellulose microfibrils in the guard cell walls is crucial for their ability to change shape and regulate pore opening. In some plants, guard cell walls can also be lignified, contain linin, or silicified. Pectic materials also contribute to the flexibility of guard cell walls. Cell membrane, the inner boundary that regulates the passage of substances into and out of the cells. Cytoplasm, the jelly-like substance filling the cell, containing various organelles. Nucleus contains the genetic material of the cell. Vacuoles, large membrane-bound sacs that store water, nutrients, and waste products. Changes in vacuolar turgor pressure are key to stomatal movement. Chloroplasts present in guard cells, enabling them to perform photosynthesis, which contributes to the turgor changes.
Stomata can also be classified based on their distribution on the leaf. Hypostomatus. Stomata only on the lower abaxial surface of the leaf, most common in mesophytes to reduce water loss. Epistomatus. Stomata only on the upper adaxial surface of the leaf, common in aquatic plants with floating leaves. Amphistomatus. Stomata on both upper and lower surfaces of the leaf, common in grasses and plants requiring high rates of gas exchange. Astomatus. Lacking stomata, found in some submerged aquatic plants. Functions of stomata. The primary functions of stomata are gas exchange, carbon dioxide, CO. O. Uptake. Stomata open to allow CO whole to enter the leaf for photosynthesis, the process by which plants convert light energy into chemical energy. Oxygen. OO. Release. Oxygen, a byproduct of photosynthesis, diffuses out of the leaf through the stomata. Respiration. During cellular respiration. Respiration. During cellular respiration, oxygen enters and carbon dioxide exits the plant cells through the stomata. Transpiration. Stomata facilitate the release of water vapor from the plant into the atmosphere. This process, called transpiration, creates a transpirational pull that helps draw water and dissolved minerals from the roots up to the rest of the plant. Transpiration also plays a crucial role in cooling the plant by evaporative cooling, similar to sweating in animals. Regulation of water loss. The opening and closing mechanism of stomata is vital for controlling the rate of transpiration and thus maintaining the plant's water balance. In hot, dry, or windy conditions, stomata can close to reduce excessive water loss and prevent dehydration. During the day, when photosynthesis is active, stomata generally open to allow C-hole uptake. They tend to close at night when photosynthesis isn't occurring, conserving water.